Hi, my name is Molly, and this is my video on the baculovirus expression system and how it's used to produce proteins. Some of this information I got from taking the class MIP 613, which some of you might have taken as well, so you will be familiar with this. So first I will give some background on what baculoviruses are. Baculoviruses are rod-shaped DNA viruses that only infect insects and replicate in the nucleus. They have circular double-stranded DNA genomes, and there are two variant types. The budding variant, which spread infection cell to cell by budding from the cell membrane, and the occlusion-derived virus, which forms in the cell nucleus and is kept in the structure called the occlusion body. Baculoviruses have been developed to be used for recombinant protein expression, and they are used in vaccine production. So now I will go over the general procedure of using a baculovirus expression system. The procedure is very long and there's a lot of steps, so I'm just going to give a brief overview, but there's a video in my references where the girl goes into depth and shows every step, and it's pretty cool. But this is my overview. So, first step is the gene of interest is inserted and combined with baculovirus DNA in a plasmid called a BACMID. And this plasmid has a promoter called POL-H, and the BACMID is transfected into insect cells using a transfection agent, and the cells will produce the virus, which will infect other cells, and eventually they'll secrete the virus into the media that they're plated on. And the point is to create more and more virus so that it can be infected into other cells to produce more protein. So the virus is filtered, collected, and stored, and then it's amplified by adding it into more cells. So then finally the protein is collected by vacuum filtration, and then it can be purified. And when using this in experiments, a mock transfection mixture that includes a transfection reagent but no DNA is used as the control. This figure is from a paper where researchers used a baculovirus expression system to produce a SARS-CoV-2 spike protein. These charts show the cell's density and viability up to 3 days after infection. The dark bar is the infected flask, and the light bar is the non-infected flask. These two pictures are immunofluorescent images of a blue tongue virus gene grown in insect SF9 cells using a baculovirus expression system. The left shows uninfected cells, and the right are infected cells. This figure is from a paper where researchers used the baculovirus expression system to produce a PCV2B cap protein to be used as a vaccine for pigs. The advantages of using a baculovirus expression system versus bacteria or yeast or mammalian cells is that it produces a high protein yield. It ensures correct folding of proteins, and the proteins remain biologically active, and it is cheaper and faster than mammalian cells. However, it is more expensive and time-consuming than using bacteria or yeast for protein expression.